Well, yeah, first, thanks, Philip, for having me. My name is Geert Vanden Bossche, and my background is veterinary medicine. Early on in my career, I specialized in virology and immunology, primarily in human diseases. That was to begin with in academia. I then moved on to industry where I initially worked in lay development, more kind of like project management, regulatory environment, moved on to research and development, where I took a deep dive at two big vaccine companies in adjuvants and alternative routes of vaccine delivery. And I then worked also in vaccine discovery with global health organizations. I was senior program officer vaccine discovery at the Bill and Melinda Gates Foundation. Also worked during the Ebola crisis in West Africa a number of years ago with Gavi, the Global Alliance for Vaccines and Immunizations, where I was the Ebola program manager. And I also worked on natural killer cell vaccines. That was my own project. So I consider myself as a problem solver, putting the pieces of the puzzle together. And that's why I'm primarily emphasizing the kind of several different disciplines I touched upon, rather than explaining the detail on the different institutions and universities where I worked. Well, Philip, I would say in a nutshell, I could basically summarize this in one sentence. What is extremely from the very beginning, in fact, worrying me is that we don't have herd immunity. So the virus continues to evolve. The virus, continues to be transmitted. And it's very clear from all the observations from the mutation spotters that the virus continues to evolve, and that this evolution has now become really a function of the immune pressure that is exerted by the population. I mean, this is largely acknowledged. This is not new. This has been published early on, that initially we had let's say, these converging mutations that were clearly by molecular epidemiologists were due, according to their analysis, were due to the immune pressure exerted by the population. And we have, even after the advent of Omicron, everybody thought, well, Omicron is going to be a blessing. But basically, after the immune system got an opportunity to catch up a little bit and to make the disease, in some cases even asymptomatic. We have seen that still, after vaccine breakthrough infections, for example, the immune system was still not capable of controlling the virus, and the virus has continued to evolve. And what we see right now, to cut a long story short, is that we have seen that the virus became more and more infectious, which was in fact pretty logical, since most of the immune pressure was exerted on spike protein, and spike protein is responsible for viral infectiousness. But recently, and that is where, once again, I was ringing the alarm bell, when I heard about mutations in GN1, this descendant from BA286, that was characterized not only by additional changes in spike protein, but primarily also by changes in other viral proteins that were, for me, very clearly reflecting that the virus is experiencing immune pressure no longer on specific epitopes of spike protein, but on its infectiousness in general. And now, you remember at the beginning, everybody was saying, well, GN1, it's highly infectious and it becomes more and more dominant. That's what we are seeing right now, that, it continues to dominate, and it's prevailing in more and more countries. But what we have seen as well is that, whereas people were initially saying, well, don't worry too much because it's more infectious, but there is no proof whatsoever that it is also more virulent. And what we are recently seeing in a number of publications is that people are now showing that due to, some mutations, for example, these flip mutations, the virus is more fusogenic. So there is more, the fusogenicity, for those who don't know, is correlated, so to say, with the virulence of the virus. And that is very, very strange, because initially nobody was saying, well, the GN1 could be more virulent. And now all of a sudden there is this findings in vitro that it is more fusogenic, which would be correlated to, more virulence. And what is very interesting is that I've always been saying that since the advent of Omicron, where we had this, remember, antibody-dependent enhancement of infection, that these very same antibodies that would enhance the infectiousness of Omicron were also protecting against virulence. Now, if you start to analyze this, for example, in vitro, so in the absence of the virulence inhibiting antibodies, you will see, of course, that you have these indications of enhanced virulence, fusogenicity, whereas you will not see them in vivo, in the field, as long as people have, high enough concentrations of these non-neutralizing antibodies. What it illustrates for me now, the fact that we have these rising rates of hospitalization and deaths in a number of countries, in many countries actually, is that most likely the concentration of the non-neutralizing antibodies starts to decline, so that now the picture in vivo is more and more reflecting what people are seeing in vitro, in the absence, of course, of the non-neutralizing antibodies, namely that we are starting to see, indications of enhanced virulence. All this taken together is, for me, simply confirming what I have been predicting already a long time ago, that once the virus would have achieved its maximum infectiousness, and according to a number of virologists, the level of infectiousness that GN1 has now achieved is really maximum. Many of them are saying one cannot imagine that the virus could even become more infectious than it is already. When that stage is achieved, in order for and the virus is still under pressure, there is still pressure on viral infectiousness and transmission, is that the virus will now shift to enhanced virulence, to, whereas, in the beginning, or whereas before, it was enhancing its intrinsic infectiousness to transmit, to increase the chances of transmission from one person to another. It's like it's now increasing its chances of disseminating within the body itself to ensure its continued propagation. So this is very, very worrisome for me, because it's simply, in fact, confirming what I've been predicting based on my analysis of the immune pressure put on the virus. And now it becomes obviously more and more reality, in a sense that, according to my humble opinion, the increase that we are seeing right now in hospitalizations and death rates is, for me, the prelude only to a massive change that the virus will undergo to overcome collectively, not just in a limited number of people, but to overcome collectively the immune pressure that is now put on viral virulence by virtue of the declining concentrations of the non-neutralizing antibodies. I think it's what people need to understand is that some of the, as Rob was pointing out, some of these immune mechanisms that are in place right now are indeed not sustainable, that can be immunologically explained. And but it's not like all of a sudden this kind of immunity is going to appear from one day to another in all of the population. So it starts to decline. That is where you reach suboptimal levels. And that is where you start to select some variants that are able to overcome the suboptimal immune pressure. And I think what may be difficult for people to understand is, why would you all of a sudden have this spectacular mutation that I'm talking about? It is because, nevertheless, when you consider, the entire population, if you consider the entire population, there will always be still a big chunk of the population that is protected, and that is relatively well protected. For example, against the transmission, against the disease, etc. And this will hamper the propagation of the virus, even though some people have already lost that immunity, and the virus can cause in a certain percentage of the population, for example, severe disease, what we are seeing right now, as Shankara was pointing out. Still, the bulk of the population is still enough protected to kind of generate a very annoying situation for the virus in terms of ensuring its survival and its propagation. And that is where the virus, just as we have seen with Omicron, will all of a sudden, really all of a sudden, undergo a spectacular mutation to make sure that now it can overcome the residual defense mechanisms that were still existing in all the rest of the population. That is what the virus needs to do to ensure its propagation. And then what is also, second point, and then I will stop, very important for people to understand, is that whenever you have an infectious virus that can cause, that can destroy cells, that is cytolytic, and many viruses are cytolytic, and SARS-CoV-2 is a cytolytic virus. So whenever you have a virus that can infect and destroy cells, and you have no longer sufficient immunity in place, as Rob was pointing out, innate immunity didn't get trained and the adaptive immunity got derailed, then every single virus, again, that can infect cells and destroy cells in the absence of sufficient immunity is per definition highly virulent. People always think, there is this aspect that conditions infectiousness, and then there is this aspect that conditions virulence, etc. If you take away the immune response, the immune defense, a virus that is infectious, living viruses are infectious, and that can destroy cells, is per definition going to become highly virulent. And that is my fear, that once we will have reached this point where the virus will have undergone this dramatic mutation that will overcome the last line of immune defense that is still in place, and that we know is not very robust, as again, Rob was pointing out, that then what we will see is just a huge wave of severe disease. Philip, the answer is no, because, what you have to think about is, and frankly speaking, I don't care which antiviral. It's the only possibility we have is to prevent the infection. But if you think about it, you will need an antiviral that, is safe, is very, very safe, right? It also needs to be widely available. Look how many countries we have been vaccinated, and I think maybe this was before our session. Shankara pointed out it doesn't make sense to do this in a few countries. You need to do this globally, and then it also needs to be made available at affordable cost. I mean, these are 3 points, 3 things we all agree upon: affordable cost, sufficient supply. It needs to be safe and widely available. We don't even need almost to talk about efficacy, but we know that is monoclonals, right? That is what the virus has been working on, is to develop resistance against those monoclonals. So obviously the other medications we are talking about have like multiple pathways where they can curtail the synthesis and the production of the virus, so to abolish productive infection. And if we have several different mechanisms that can be combined and do this, I think in terms of efficacy, we have a much higher chance of being successful. But don't forget the practical limits, the availability, the price, and the safety of the antiviral drug that should be used to tackle this, massive, widespread, large-scale problem. Well, Philip, I had some kind of warning. I've been talking about this multiple times. A small-scale warning like 8-9 years ago with the Ebola crisis, but it was very, very similar what happened there. And, but it was small scale. So, but even there was no reaction, was no feedback, there was no, acknowledgement of mistakes and, people wanting to correct the situation and doing something different. It's all about large talks about, pandemic preparedness and all this type of things. So, when this thing came, that was the reason why I was so self-assured to immediately ring the alarm bell, because I knew that this is the way things happen, and that is how it is, managed by even the scientific medical community, etc. So I don't know how we are going to change this, how we can change this. I personally also disagree that there are 25 different approaches to the science of such a complex problem. I'm the first to say, okay, we had to open the debate. Nobody engaged really in a debate. But on the other hand, I also don't think that there are, as I was saying, 25 different correct approaches. Even if you see what we are talking right now, it seems like we can streamline this and we can narrow this down to very, very clear and very, very simple recommendations, right? So this complexity is something that looks to me very, very artificial from day one, in fact. And where I must say I'm not surprised, because that is the way these pharmaceutical companies and if you start making these things, this infrastructure and the hierarchical structure very complex, you end up with some very, very complex solutions that are not really solutions. I was not surprised, and I knew from the very beginning that even though we would cry and shout it from the top of the roofs, that there was a high likelihood that it would not really have an impact. But now the situation is different, because I think right now that there is a lot of people on the other side that are really panicking about what could possibly happen, because it's very clear if I look right now at the different publications, etc., that there is a number of highly ranked scientists and experts that are no longer ruling out that we may be facing a very, very worrisome and difficult situation with an unpredictable change of the virus in a direction that we don't want to see, in fact. And there is no plan B. That is the problem. There is no plan B. We know that just masking up and lockdowns is not going to be a solution. And we also know that continuing, the vaccination will be very challenging as well if you are facing a very acute situation. Yeah, well, I very much agree with, with regard what Rob and Shankara was saying with regard to the medical community, but I would also like to call on the scientists, because their insights and what they are publishing also has a tremendous influence. And what I've been seeing is that for many scientists, this has been just a huge opportunity, to make publications and to tease out all kinds of molecular details to make yet another publication. And publications are very, very important. But when you're dealing with an acute situation, as the one that we have been experiencing and that we are still experiencing, I think they have also a very important responsibility and a very important task in, consulting together in order to make, for example, also models that are reliable, that are, taking into consideration several different aspects and several different disciplines. That has not been done, and I think they also have a huge responsibility. And I still hope that they can help us, to shift gears. But as you were pointing out, Philip, and that's my last point, it is, more than time. I don't know if it is still time, because I just wanted to point out, when we have seen this massive wave of Omicron breakthrough infections, remember, it was preceded by sporadic vaccine breakthrough infections due to previous variants. We saw a number of vaccine breakthrough infections already with Delta, etc. And then all of a sudden, we had this huge wave of vaccine breakthrough infections with Omicron. This is similar to what we are seeing right now. We are seeing already a number of virulent vaccine breakthrough infections, but not a massive wave. But it is the prelude. And I think with regard to what Shankara and Rob were saying, treating people in a prophylactic way with antivirals, ivermectin, hydroxychloroquine, I think we need to do it now. I don't think personally that we have still time to wait. And it would, of course, be fantastic if we could convince leading authorities of this, the need to do this and to act right now instead of keeping talking. Over.